Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar, titled Optimizing Lentiviral Purification, Enhancing Chromatography with Monoliths. I'm Roisin McGuigan, an editor at BioInsights, and joining me today is Luka, who will explore how Monoliths streamline lentiviral vector purification, doubling infectious recovery, cutting development time by half, and reducing costs with a scalable, efficient, and reusable approach. After the presentation, we'll have a live Q&A session. So feel free to pose your questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to them during the session. Now, I'd like to introduce our presenter. Luka, with a PhD in molecular mechanisms of disease, specialises in downstream process development for lentiviruses as a project manager at Sartorius BIA Separations. So, without further ado, I'll hand over to Luka, who'll kick us off with today's presentation.
Thank you, Roisin. So I will first give you all a brief introduction to the Sartorius BI separations and our monolithic chromatography. And then I will present to you the case study on the lentiviral purification optimization before finishing with a summary. So first, a little bit about the Sartorius BI separations. We are based in Slovenia, which is a country that is bordering Austria, Italy, and Croatia. And more specifically, we are located in Ajdovština, which is a region famous for its wine. We are a monolith experts and exclusive producers of CIM, or convective interaction media monolithic chromatographic columns. We have over 25 years of experience with the columns, and we have over 250 employees. We are specialists in the purification of large biomolecules and have significant experience with different large biomolecules such as AAV, adeno, flu, pDNA, mRNA, LNPs, VLPs, exosomes, and more. And we are Sartorius Center of Excellence in Gene Therapy, and we offer solutions for downstream process development, manufacturing, and analytical method development. We offer a comprehensive monolithic chromatographic portfolio, and we have solutions for both preparative chromatography and analytical chromatography. On the preparative side, we offer screening chromatography solutions such as the CIM multi-well plates, the example of which you will also see presented today. And we also have the Simultus preparative columns, which are scalable formats from 1 ml to 40 liters, are ready to use, and are multi-use chromatographic columns. On the analytical side, we have the CIMAC analytical columns, which offer high resolution in short analysis times. And we also offer a PATFIX liquid chromatography system, which can be used for in-process control and product release quality control. And for all our products, we also offer the Cornerstone Biomanufacturing Development Service, which is a service where we can develop preparative and analytical chromatography. So what is a monolith actually? So monoliths are actually single unit structures with highly interconnected convective channels. These channels enable that the flow through the monolith is laminar, which means that the shear forces on the samples that get pumped through the monolith are low. And this makes the monoliths ideal for large molecule applications. As mentioned, the monoliths are scalable from milligram to kilogram production and are CGMP compliant. They offer higher resolution applications and enhanced productivity. And as mentioned, they come in pre-packed columns and are reusable and alkaline stable. So how does the chromatography with monoliths look like? So we have our sample with our molecule of interest, which is pumped through the monolith and under the conditions which enable the binding of the molecule of interest. So in our case, this was a virus which attaches to the binding sites that are distributed throughout the monolith. While the virus binds to the sites, the impurities pass through it. And once the impurities are washed out, the conditions in the mobile phase are changed to enable the elution of the captured virus particles. And these released viruses are then collected as purified target molecules. And now I will present to you the case study with the monoliths on the lentiviral purification optimization. So first, just to give you a brief background on the lentivirus. So lentivirus is a retrovirus, meaning it has an RNA genome, or specifically it has 2 copies of RNA genome. It's about 80 to 120 nanometers in size, and it has a large capacity for inserts, up to 9 kilobases. It is capable of integration in both dividing and non-dividing cells, making it a very suitable vector for cell and gene therapy. Currently, it is mostly used in cancer treatment applications, primarily in ex vivo, as an ex vivo gene delivery. The downstream purification of lentivirus, however, still remains one of the bottlenecks for its more widespread use. And our goal was to basically see if we can tackle this challenge by using monoliths for the downstream purification of lentivirus. Our initial experiments were focused on testing different chemistries where the QA proved better over other chemistries. And to try and improve the downstream conditions, we decided to try and optimize buffers for the downstream process with the loading of clarified harvest. However, as you can see on the right, the initial experiments didn't seem to result in significant improvement in the downstream process recovery just with the changing of buffer and buffer compositions alone. Basically, we realized that we need to change the loading conditions as well to get a better recovery. And we need to do this in a systematic way to improve the downstream recoveries. That is why we decided to use the design of experiments approach. So the design of experiments was designed with different parameters that we found interesting to evaluate for the downstream process. And these were selected in 2 groups. The first was so-called initial conditions, such as the pH, the NaCl, and saccharose concentrations. And then in the second group were the different additives that we wanted to test, such as the magnesium chloride, the paloxamer 188, and amino acids. The main concern with the lentiviral downstream process for us was the lentivirus infectious recovery. And as such, this was the only readout that we were focused on during the optimization process. And any additional characterization would then be performed once the final conditions have been set up and reference run with the established conditions performed. The testing of effects of different parameters, however, is made difficult by the fact that there are large variations in the infectivity between different experiments. As such, replicates of the same conditions would be required to get more reliable results. And in order to try and increase the throughput and reliability of the results, the experiments were performed on the SIM-QA 96 well plate, which you can see on the right. And we performed the experiments in multisets and in parallels. So as mentioned, the goal of the DOEs was to improve the loading conditions under which we would get better downstream process recovery. However, the loading conditions are difficult to adjust in the clarified harvest. As such, we decided to simplify the process by mixing the clarified harvest with mobile phase A. And we adjusted the conditions in the mobile phase A. So the conditions that you see on the right are the conditions that have been used in the mobile phase A. So if we look at the initial results, so as mentioned, we looked at first at the pH, the saccharose, and the NHCl effect. And if we look at the coefficients at the bottom right, we can see that of the three parameters, the NHCl seemed to be the most important when determining the recovery in the downstream process. Specifically, the increase in the NHCl concentration led to a better recovery in the downstream process, while the effects of pH and saccharose didn't seem to have a significant effect. And you can also see this reflected in the contour plots at the top right, where you can see that the best results, which are in the red, are achieved at the highest NHCl concentration, regardless of the pH and saccharose concentration. And based on these results, we decided to do a follow-up DOE where we adjusted the conditions according to the results. So we increased further the NHCl concentration, and we also used a wider range of pH and the saccharose concentration to try and determine if they have an effect as well. So if we look at the results from the second DOE at the bottom right, we can see that the NHCl concentration was again a very important, significant factor when determining the downstream process recovery. But this time, the pH was also a significant factor. While the saccharose, on the other hand, again, didn't seem to have a significant effect on the downstream process recovery. And if we look at the contour plots, what we can observe is that the best recoveries were again achieved at the highest level of NHCl, while the pH has to be the lowest. So lowest pH, highest NHCl was the best recovery, while the effect of the saccharose didn't seem to be significant. So based on these results, we decided to perform some additional testing for the pH and NHCl concentration until the final conditions were determined. And for buffer A, these were the 50 millimolar BTP, 500 millimolar NHCl, and a pH of 6.75. And following this, we then also decided to perform DOEs for different additives. So the first DOE was performed for the addition of magnesium chloride and poloxamer 188. So the results, as you can see on the right, if you look at the coefficients plot, what we can see is that the addition of magnesium chloride seemed to have a beneficial effect on the downstream process recovery, while the addition of poloxamer didn't seem to be significant. The addition of both magnesium chloride and poloxamer, however, seemed to have a negative effect on the downstream process recovery. And these results are also reflected in the contour plots, which you can see at the top, where the best results, the best recovery in the red, was again achieved at the highest level of magnesium chloride addition and the lowest addition of poloxamer. The effect of magnesium chloride addition, however, seemed to still be in the range of error. And as such, we decided to also perform a follow-up experiment where we confirmed that the addition of magnesium chloride had a beneficial effect on the downstream process recovery. And following this, we also performed DOE for the addition of arginine and histidine. If we look at the results from this DOE, if we look at the coefficients, what we can observe is that the addition of arginine again seemed to have a beneficial effect on the downstream process recovery, while the addition of histidine was irrelevant. And you can also see this reflected in the contour plots, where the highest level of recovery was achieved with the 10 millimolar arginine addition. As the effect of arginine was also inside the range of error, we also confirmed the beneficial effect of arginine addition in a separate follow-up experiment. So at this point, we were closing on, or we got around 90% infectious titer recovery. So at this point, we were quite satisfied with the conditions that we got, and we selected a final buffer formulation, which was the following. So for the buffer A, we had 50 millimolar BTP, 10 millimolar magnesium chloride, 10 millimolar arginine, half molar NaHCl, and a pH of 6.75. For the buffer B, or the elution buffer, the conditions were the same, with the exception of 2 molar NaHCl. So these final conditions were then tested in 2 confirmation runs, which were performed using 1 milliliter 6 micron simultus QA column. So you can see one of the chromatograms, so a chromatogram from one of the runs here on the right. So we performed this chromatography experiment with conditions that could be also scalable to larger volumes. And if we look at the chromatogram, we can see that majority of the impurities, or the UV signal that you can see, was in the flow-through, while in the elution, we got a nice single peak where our virus was eluted. And when we analyzed these samples, we looked at the infectious recovery, and in the 2 runs, we got the average 70% infectious recovery, which was an over 3-fold increase compared to the start. We also performed some additional analysis with such as RT-PCR and particle recovery, and we observed 40 or 46% average recovery with the 2 analytics, which actually indicates that during the process, we also removed some additional incomplete or damaged capsids, while still retaining majority of the infective virus. We also looked at the protein impurity removal, and we observed that we removed over 3 logs of host cell proteins. So we had a very high protein impurity removal. And when we looked at the capacity, we determined it to be around 2.9 E12 particles per milliliter of monolith. And that is on a 6 micron channel QA column. The results from the RT-PCR and video drop, so particle recovery, as mentioned, indicated that we could remove some impurities, some particle impurities during the process. However, we were also interested to look a bit more into what the particle composition of the different samples looks like. As such, we decided to use Leprechaun, which is a machine that basically enables the determination of particle compositions of different fractions. So how it works basically is it works kind of like an ELISA, where you bind the particles to a wafer, a chip, and you stain them for different antigens. So in our case, we were interested in the presence of VSVG, the P24 capsids, and RNA. And if we look at the results on the right, so we have the average composition from the two runs of different fractions. And if we look at the composition, so in the yellow, we have the so-called complete lentiviral particles, which have VSVG, P24 capsid, and RNA. And the rest are particles that are either missing the RNA or the capsid or both. So if we look at the composition of clarified harvest, we can observe that initially we have about 1/3 of the samples of the particles which are complete lentiviral particles, and the rest are incomplete or empty. And if we look then at the composition of the flow-through, what we can observe is that the majority of the particles are actually lacking both capsid and RNA. We have a very small amount of particles that are actually complete in the flow-through, which indicates that during the chromatography process, we managed to eliminate a lot of incomplete or empty particles in our flow-through. And again, if we look at the composition of the elution fraction, what we can observe is that the over 50% of the particles in the elution are positive for VSVG, P24 capsid, and RNA. So are complete lentiviral particles, which indicates that during our downstream process, we can successfully enrich for the full lentiviral particles while removing the incomplete or damaged empty lentiviral particles. So with that, I would like to summarize our conclusions. So we have shown that the QA monoliths are a suitable tool for the lentiviral purification. We have shown that using the 96 well plates and the DOE approach, we successfully optimized the lentiviral chromatography process and got increased the infectious recovery from 20 to 70%. And we have also shown that during the purification process, we successfully enrich the full lentiviral particles while removing other particle impurities. Yeah, of course, the work that I presented was a lot of people have contributed to it, and a lot of parties. And of course, I would like to thank all of them for their contributions. Yeah, I would like to thank everyone for listening, and I'm happy to take questions. Thank you.
Well, thank you, Luka, for a great presentation. And now we're going to start our Q&A. We've had lots of questions in from the audience. Please do keep them coming in. We'll get to as many as we can, and any others we will answer via email. So, Luka, if you could just pop your camera back on and join me.
Yes.
Oh, fantastic, thank you. Right, so I'll get us started with our first question today. So, what lentiviral pseudotype was used?
We use the VSVG pseudotype in our experiments.
Fantastic, thank you. Next question here. Is only the QA column recommended for lentiviral downstream?
Well, so the QA column performed better in our case, but there are plenty of publications also on the DAE columns. So I would actually suggest for anyone interested in the lentivirus to basically test the two columns for themselves and basically determine which works better for them.
Absolutely, thank you. Next question here. Why was lentiviral downstream optimization performed on a 96 well plate?
We used the 96 well plates because because it offered several advantages. So basically, first of all, we could reduce the amount of material that we required to perform the optimization, and the buffer consumption was lower. Secondly, it enabled us to basically do the experiments in multisets and in parallels, which basically means that we could perform an entire DOE in a single run, and we could also perform it in duplicates or even triplicates. And that way we could increase the reliability of the results that we got. And of course, it also, when you do everything in single day, it also reduces the day-to-day variations in the experiments.
Great, thank you. Great answer there. Question from the audience here. In screening chromatography, such as a 96 well plate, before screening, should we try flow chromatography once with gradient for checking the peak?
I would suggest yes, because you still need, basically on the 96 well plate, we had what you would call a step elution, because you can't really do a gradient. So yes, I would recommend doing a gradient on a chromatography machine, and basically determine which conditions are required for the elution. So that you can then use it during the purification, during the optimization process on the plates.
Great, thank you. And another question here. Is the clarified harvest starting material coming from an adherence production platform or from suspension? And if adherence, what is the impact on the study of the serum?
We had a suspension cell culture, yes, in which the lentivirus was produced.
Okay, great. Short and sweet answer there. Thank you. Next up, what cell type was used for infectivity analytics?
We used the HEC293 T cells for the infectivity, and we had a GFP reporter.
Fantastic, thank you. Another question here. What are the advantages of using design of experiments?
The design of experiments basically enables you to test conditions in parallel. So basically, you could determine how different parameters affect the downstream process at the same time. And basically, it enables you to distinguish which parameters are important and which ones to focus on in a very structured way. And you can get a lot more information from a fewer experiments that you would get otherwise. So for us, when we were not really sure which parameters to focus on, it was a very, very nice way to basically systematically determine like how important, let's say, NHCl concentration was in the loading conditions for the downstream process, which without a structured approach would be a bit more difficult to do.
Fantastic. Yes, so some great benefits there.
Yes.
Great. So next question, does it matter whether you use a 2 or 6 micron channel monolith?
So we use the 6 micron channels just because we were thinking on the if someone would go large scale. Like on larger scale, you would prefer to have faster process times. And for faster process times, you would prefer to have a 6 micron channel column. On lower scale, on small scales, I don't think, or as far as I know, there is no significant difference between 2 or 6 micron channel columns. Obviously, 2 micron channels columns would also have a significantly higher capacity, even compared to 6 micron. So they will still be useful for small or even medium scale applications. But for large scale, I think you would probably require 6 micron channel columns. And that is why we were working with 6 microns from the start.
Great, thank you. Next up, this is a slight tongue twister, so bear with me. Which is better, DEA or Q in AEX for lentivirus?
Well, I think, as I mentioned, in our hands, the QA performed better than the DEA. But as mentioned, I would suggest for everyone to basically test them out for themselves, because we do know that the lentiviral materials can be different from one producer to another, from one batch to another. So yeah, I would suggest that they test them from themselves. But we have the evidence that it works both on QA and the EAE.
Absolutely, thank you. Next up here, I've got a question for you. What's your sodium chloride concentration in your clarified harvest load material?
To be honest, I'm not quite sure. So basically, we do know that there is conductivity in the clarified harvest. We don't know the exact composition of the clarified harvest, because obviously there's media in it and so on. But that is why basically we decided to adjust the conditions in the buffer itself, which we know we can control. And that way we could then control the conditions in the loading material. But yeah, the clarified harvest itself has a lot of different ions, different impurities, different additives. So that's why we decided to basically not tamper with that. It's also much easier for any other process than if you want to basically replicate it, to basically just say, okay, we will just use this buffer and we will just dilute clarified harvest in it. And yeah, it's much more simpler that way.
Absolutely, thank you. And next question for you here. How were the LV samples stored?
So in the elution fractions that had high conductivity, we diluted them first with buffer without salt, so some storage buffer, and we froze the samples in the -80. So yeah, we were storing them in -80, and we tried to make sure that the conductivity of the samples was not too high. So
great, thank you. And another one here. Do you offer any training courses on the use of the monolithic columns?
Yeah, I mean, there are definitely people that are available to basically go to the site and offer a training course for the use of monoliths. We also have plenty of, I think, material on how to use. It's pretty simple, basically, because it's plug and play. So you basically just, screw the column on the ACTA or like any other chromatographic system, and basically follow the procedure there on the for the preparation of the column for the chromatographic process. But yeah, we do offer also, of course, training on site, and we also have we can also offer training basically if anyone would come to our site.
Great, thank you. And another question here: was BTP the best performer in the initial buffer screening?
BTP seemed to be quite comparable to some other buffers. But it had a larger pH buffering range. So for our application, for our testing, we decided to use BTP due to the pH buffering range. But when we looked at the how well it performed compared to other buffers, it seemed to perform quite well, or comparable at least.
Fantastic, thank you. And another question here: were the lentiviral vector samples cleaned up prior to loading onto the QA column? And if so, can you outline how?
So basically, we just had a clarified harvest, and no other cleaning or other processes were involved before the application to the column. We just basically took the clarified harvest, diluted it with buffer A, or the mobile phase A, and applied it to the column, and that's it. No other processes were involved.
Great, thank you. Another question here: why did you try to use saccharose?
We tried different sugars, different additives in the beginning, and we didn't see a significant difference between them. So basically, saccharose is something that's very uniformly used for stabilization of different viruses, and as such, it seemed also like a good candidate to try and use in our DOE experiments. And we also figured that if there will be an effect of sugar addition on the lentiviral stabilization, we should be able to see it with the saccharose. Of course, there might be differences between different sugars, but if you don't see any effect with saccharose, there are lower chances that you might see it with some others.
Mm-hmm, mm-hmm. Great, thank you. And next up, will the recovery by RT-PCR always be higher than the recovery by infectious titer?
Our recovery on the RT-PCR was lower than the infectious titer.
Oh.
So yeah, we usually get lower recovery on the RT-PCR than the infectious titer, which is what is very interesting, because this is what actually made us think that wait, there's something else going on here, right? So we get the infectious particles, but not the particles that might have RNA inside, So that means that we are removing some particles which are not infectious, but still have RNA. And that is why we then used the leprechaun to basically determine the sample composition, and where we then also confirmed that we are enriching for the full lentiviral particles.
Great, thank you for clarifying that one. Now, this next question is rather lengthy, so please bear with me. 2 molar sodium chloride seems like a lot for lentivirus. So do you have to dilute the sample immediately post-dilution? And to what sodium chloride concentration? And then the last part is, do you also add any cryoprotectant like sucrose or glycerol?
So 2 molar sodium chloride concentration is the highest concentration, but we use a gradient for the elution of lentivirus. So this is not a step elution, it's a gradient. So obviously the concentration of NaHCl in the elution is lower. And we dilute the lentiviral harvest, sorry, the elution, yes. And we diluted it basically accordingly to what we observed to be the best concentration. Basically, we try to do a couple of tests and basically just see what the difference is. So I would actually recommend for people to basically try that out for themselves, what concentration it would work for them. And when regards to the cryoprotectants, no, we do not have cryoprotectants that are like glycerol or anything like that. And we use a storage buffer, which is basically a very simple buffer. I'm not sure I can disclose what it is, so I would prefer not to. But it's basically some simple buffers without salt.
Okay, no problem. Thank you very much. Next up, how did you check empty and full particles for the lentivirus?
Basically, as I mentioned with the leprechaun, we can see the composition of the particles, and we basically empty full is basically kind of how would you say, what is the terminology that is used with AAV, but basically it's what you would consider a complete or incomplete particle. So if the particle had VSVG, P24 capsid, and RNA, then it was a complete particle. If it didn't have all three of them, it was an incomplete particle. And basically, this is just to make it easier to basically distinguish between what is important and what wouldn't be technically important. Because we, of course, assume that all the infectious lentivirus is a complete particles.
Fantastic, thank you. And one in a similar vein, could you explain why full virus is concentrated?
Well, we believe that basically the full virus would have a bit tighter binding to the column compared to an incomplete virus. Exactly why that happens, I'm not exactly sure. There might be some basically when the budding out happens from the cells, a complete virus might bud out a bit differently than a virus or like some, let's say, RNA without a P24 capsid. Because of course we know that the cells also produce exosomes and other particles, other lipid particles, while the lentivirus is produced as well. So we know that there are other impurities in there. And they most likely have other post-translational processes or have a different lipid composition, lipid membrane composition compared to a full lentiviral particle. So I believe that, or I assume that basically this composition is then what determines how strongly the particle binds to the column, and if we can then basically wash it out during the process.
Fantastic, interesting, thank you. Next up, I have a question here. Have you tested the stability of the virus with a high sodium chloride concentration, i.e. over 500 micromolar? And was the pH and sodium chloride adjusted before loading the clarified harvest onto the Smeltis QA?
I'm not sure what kind of question, what exactly is the question with have I tested the stability of the virus with different NaHCl concentrations? I mean, so we dilute the harvest with the buffers that have 500 millimolar NaHCl. So obviously we increase the NaHCl concentration in the loading material. I wouldn't. So I would expect that if we leave it for a longer time with a higher level of NaCl, that the infectivity will drop. We can also observe this if we basically when we freeze a what you would call a loading material and clarified harvest, we can usually observe higher infectivity on the clarified harvest. So I would say that yes, the higher if you leave antivirus for a longer time on the with the NaCl, higher NaCl concentrations, that the infectivity would drop, which is also why we dilute the elutions. And I forgot what the second part was. If the pH and NaCl was adjusted before the loading, we adjust them only with the addition of buffers. So we didn't do any other adjustment of the pH or NaCl. We just use the buffers.
Great, thanks so much. Great answer. Next up, did you use a TFF step before AEX to exchange into the correct high salt buffer?
No.
Okay, another short and sweet answer there. Fantastic, thank you. Next up, did you try chromatography in ACTA not using MALS?
No, because MALS is what you want to see. Because UV is not does not indicate the virus necessarily, at least. What we could observe sometimes is a UV peak, which basically was empty of lentivirus. It's basically just impurities. So it's actually MALS that you should follow, not the UV. So obviously, MALS gives you just particles that are larger in size, so it's not lentiviral specific, but it's much better than following the UV.
Great, thank you. Next up, do you have data that shows scalability to larger bed volumes?
We didn't do specifically larger columns than 1 ml, but basically we ran with the conditions that can be transferred to a larger columns. So the problem is that we have the problem. I wouldn't say it's a problem. Basically, is that we have the material we have is limited. And we wanted to basically show that we can optimize the process, and we did optimize the process. And it's actually we have quite a high capacity, so we would actually require basically more volume than we had basically altogether to try and go to a larger column. But we did basically use the conditions that are that can be transferred to a larger column. Yes.
Great, thank you. Next up, do you see that residence time during chromatography has an effect on infectious recovery?
Yes. That is why we also believe that we have seen basically that when we did the optimization on a plate, we could get 90% or even higher recoveries, And then once you go to a chromatography on 1 ml column, and basically you use the conditions that are used in chromatography, obviously you can't get the processing time as fast as on a plate. And this most likely affects the lentivirus infectious recovery, which is also why we got average 70% infectious recovery, which is still more than than most others publications have shown so far. So yes, I would say residence time is quite important, but at least based on what we've seen, the conditions that we developed on our columns are very suitable for even if our residence time is a bit longer.
Great, thank you. And next question I have in here. Do you do any dilutions of the clarified harvest before loading?
Yes, as explained, we dilute with the mobile phase A. I would actually recommend that people basically determine what dilutions they would perform. We performed the dilution 1-to-1 with clarified harvest to the buffers. But of course, I would recommend that anyone would basically test them if that works for them, or if they would need more dilution or higher dilution than that.
Great. Thank you. I have one more here. Well, this isn't actually a question, but more of a comment. Wonderful session. Congrats and thanks. Thank you very much. The audience members sent that in. We're delighted you enjoyed it.
Yeah, thank you.
So thank you, Luka, for answering what was a really mammoth number of questions. I'm afraid that's all we've got time for today. Any questions we weren't able to get to, we will reply by email. This webinar will be available on demand tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with feedback on today's webinar, the link to which can be found in the resources on the right-hand side of the screen or in the on-demand email. So all that's left now is to thank Luka once more for a great presentation and a really great Q&A session. And thank you to our audience for listening. We hope you'll join us again soon.